So this was a very nice session, and I'm really happy now to go to question and answer with Maria Musaji, and I'm sure she will have a lot of questions, and I hope all of you just send some questions.
Hello, everyone. Just thank you so much for just the wonderful talks in this session. And I just want to welcome Professor Julie Daniels, who's a professor of stem cell biology at the Institute of Ophthalmology, my close colleague, and Veronica back to this panel discussion. I've received some questions through the chat. So the first one I'm going to direct to adults. This is from Elizabeth Simpson. Have you derived isogenic control iPSCs?
No, we haven't yet. It's an option we considered as well. So for now, we are using iPS that we generated from controls, so people without aniridia, and then we're growing them in parallel and compare directly. But the isogenic lines would be the ideal control, yes.
Yeah, may I add a comment on that? Mm-hmm. I think the best is to use the patient-derived iPS and to use CRISPR-Cas9 to rescue the mutation, to correct the mutation. It will help to have the same epigenetic background, and that would be the best.
Absolutely, I agree. David, yeah, David, a couple of questions for yourself. So, what would you predict to be the clinical phenotype that one would expect from that increased inhibitory effect of neurons secondary to the PAX6 mutations in patients?
Okay, so there's a problem with the assumption behind that question, and I have to take responsibility for not including everything that we did in my talk. I think you have to be careful when extrapolating from the mouse work that we've done to human patients. Very careful. First of all, we did this study because we want to know what PAX6 does, which is obviously relevant for understanding the human condition, but it doesn't directly translate. So, for example, what I didn't tell you is that if we follow these cells as they subsequently develop, most of them eventually seem to die. So although they're making aberrant inhibitory neurons at this early stage, later on in postnatal life, they seem to collect together into these balls of cells underneath the cortex and vanish. So the result of that is that you actually end up with a cortex that's smaller than normal, but it has a normal balance between the excitatory and the inhibitory. So it's these cells should have made excitatory cells, but they didn't. And now there's something wrong with them. Okay, so they're not making inhibitory cells that can become a functional part of the brain. So we wouldn't imagine that there will be a phenotype necessarily, that would be associated with an overproduction of inhibition in the brain. So that I apologize, because that's probably going to be confusing to people now who are listening, because during my talk I was talking about the overproduction of inhibitory cells, but now I'm adding in this fact that but they don't make it for very long in postnatal life. So I'm not sure that I have any predictions of the type that question is looking for. Does that make sense?
It does make sense. And I may change the next question from the audience to more of a statement. So somebody has commented that some aniridia patients seem to exceed in areas of development at a young age, in areas of logic, language, memory, and language. Oh, we said language. The question was raised maybe to everyone on the panel: has there been a study looking into this or any other possible explanation? And again, we may have talked about this, but we obviously see a lot of patients with ADHD and autism as well with aniridia, and potentially is that all linked to this? But after what you've said, the answer is we're not sure yet.
Yes, that's a pretty difficult question to answer. I mean, I did see the question actually, and I think you'd have to define what you mean by exceeding. I guess it's a bit hard to predict. I suppose my idea of what PAX6 is doing would leave open the possibility of unpredictable effects, because what I'm saying is that these cells are more open to being changed in ways that might be surprising and might be unpredictable. So I guess, I was talking about likelihoods, not certainties. So probabilities of this happening versus that happening. I mean, normally as the brain develops, what happens is that cells narrow their ability to do things, their potential or their competence gets narrower. And that's really to keep them, if you're thinking about you're corralling billions of cells to make a brain, you're corralling them. You can't be telling each one exactly what to do. You've got to put limits on what they do and then hope for the best, if you like, which is a pretty crude way of putting it. So I guess that if PAX6 is lowered or not there in the case of the model we're using, that might open the possibility for some quite unexpected things to happen in some cases. So that's about as far as I think I would go in answer to that question.
Okay, thank you. I've got a question for Daniel. Daniel's already answered a question about the concentration of duloxetine and ritanserine that restored PAX6 levels, and that's around 1 micromolar in vitro. But Daniel, looking to the future for when this could move to potential clinical trials, what would be your kind of primary clinical trial outcome measure? And also, what age do you think it would be best to maybe start using such a drug?
Well, I'm not sure I'm the good person to answer. Maybe Dominique can answer better, but I'm sure that childhood, before the onset of start of opacification, would be the best. But it's not easy to treat children. So I will leave Dominique to answer, because we have a lot of discussion on that issue, actually.
Well, so that's for sure, that's always complicated to go from pharmaceutic trials to directly patients' clinical trial, which is completely different, of course. And so we know how complicated it is and how also expensive it is. So that's always a long way when we go from the molecule to the treatment of the patient. And first of all, we need to test if it's not toxic on the ocular surface, and so that's really important first. And if we have good proof of this, we can go to some very, very few patients to compassionate study, and after that, if it does get good results, we go to a clinical trial. But at the time, the clinical trial is very expensive. So I know that Sherry Gregory Evans know that very well. So we are not at this level, but we are thinking of it really strongly.
We did last year, a similar approach on the skin disease. It was a compassionate care, actually. So now we prove that the molecule is working. It's easier to put topically on the skin than on the eye. It's less risky. But then it's more complicated to jump to the clinical trial. But we are really motivated to go forward.
That's great. I'm just going to ask Simon a question here, because I think, he really represents the drive that patients have from pushing these therapeutics from bench to bedside. But I think a lot of patients will be sitting in the audience, and I'm going to talk as a mother of children myself. When your child is given a diagnosis, and you're with the consultant, there's no doubt that you have that trusting relationship with them, and they're giving you the diagnosis and the advice and what the management will be, etc. For me, I may go away and just accept that. It wouldn't even occur to me to maybe go back and talk about a research project and how I could make a difference. So you've done that. You've taken that step to reaching out and actually meeting people and designing a project that would help your son. So how do you switch from kind of accepting a diagnosis to pushing forwards into getting a project set up and having that drive in your mind, Simon?
To be fair, I don't quite know how to basically answer that question, because I just did. My wife and I sat down and went, well, how can we help? What can we do? And, normally with anything that normally goes wrong with your child in any way or form, there's the natural path, right? There you go to the doctors and someone says, okay, so you need to give this medicine or you need to be able to do that. When you're told, well, there isn't actually anything to be able to do, and then you ask about probabilities of remaining of eyesight, and you get again told, well, we're not 100% sure. Well, it's just come down to a very common fact, which is that 1 in 100,000 people have this. So it's not the fact that it's not solvable. It might not be solvable, but not enough. I mean, you guys all have basically done that, and I thank all of you for basically doing this. But for me, it was, well, surely there's got to be something that we can do, and we might fail, and by failing we'll learn, and then we'll keep on basically going. And so for me, it's trying to be able to ask favors of as many people as I possibly can, to be able to try and hopefully basically find some answers. So I'm not sure I've really answered your question, but
well, what advice would you give to the viewers, other patients?
I mean, I would say speak to Aniridia Network hugely, or the European equivalent, because that's what helped us to be able to initially be able to do that. And the other thing is by going via Aniridia Network, you'll get to the answer basically quicker than otherwise. Because when we first basically approached Moorfields, and I'll be honest, we didn't necessarily go in the right direction, and it took us a little bit of time to be able to find you, Maria. And that is very, very helpful. And that's why I'm going to be, after this conference, I'm going to be speaking with James, and we're going to work out how we can basically work together to be able to provide the right information. I mean, we've spoken with things whereby what we want to look at going forward is a prospective study. That will require huge amounts of involvement from patients. And the idea behind a prospective study, and you will speak more eloquently than I have on this, Maria, but is that we need to be able to do tests on a number of different basically patients, be able to find markers, so that should we find a drug, we'll know what to be able to basically test for. And for that, we will need everyone within this community to be able to basically help. So it's not just funding that we will need every basically patient to be able to get involved. And so, that's from parents of children that, you need to put if you wish to put your child forward, I mean, to be able to basically help in that. And for older basically patients themselves. So I think for me anyway, I'm going to be collaborating hugely with Aniridia Network and trying to be able to ensure, which I haven't done as yet, that what we're doing, that information is spread via Aniridia Network, and that people know how they can basically help in many different basically ways.
Thank you. We have 2 minutes left. So a really quick question, Daniel. I know you have a question for David.
Yeah, thank you. David, how do you explain that the sensitive cells are on the line, a very precise line in your labeling? We can see picture. They are not distributed.
Oh yes, okay. Yeah, I get it. Yeah.
Randomly.
Okay, so I think the answer to that is that's a very interesting question, actually. Thanks. So during normal development, the cells that I described as the immigrant cells, those are the inhibitory interneurons, which normally, in a normal brain, they migrate into the cortex. They take that line, that exact line that you're seeing, okay? Now, those cells that are coming into the cortex actually happen, and this is what put us on to the sonic hedgehog. They produce sonic hedgehog. And so we had this idea that they were triggering the cells to change as they moved in. So the inhabitants, if you like, of the cortex were being triggered to change by the proximity of the immigrant cells. And we actually tested that by blocking those migratory cells, and we found that also prevented this switching, and that we then hypothesized that it might be sonic hedgehog, which they produce, that was doing the triggering. So yeah, great question, and thanks for that. I hope that was clear.